So hello again. Our class today is about how to measure mitochondrial bioenergetics within intact situations. So measuring mitochondrial function in whole animals, in tissue slices, or intact cells. And I'm mainly going to focus on intact cells because that is the protocol which we most see in the literature in which mitochondrial bioenergetics is going to be studied. Now, obviously, this is different from studying mitochondria as isolated organelles or studying mitochondria within permeabilized systems, as we saw before, because the cells are going to be treated with media that's extracellular media. When we looked at permeabilized systems, we talked about how you had to use media that was similar to the intracellular environment. Now we're talking about systems that have an intact plasma membrane, and therefore you have to use media such as an extracellular environment. So media that's low in potassium, high in sodium, and basically similar to culture media. And very often, culture media is what you're going to study mitochondrial bioenergetics in situ with. Another characteristic when you're studying mitochondrial bioenergetics in situ is that all the drugs that you add must be permeable to the plasma membrane in order to reach mitochondria and affect mitochondrial function. This is in general not that difficult to do because most mitochondrial regulators and toxins are substances that are lipophilic because they affect the mitochondrial inner membrane, and therefore they do permeate the plasma membrane. So you can use those drugs such as oligomycin and FCCP and rotenone and antimycin that we already spoke about when we were talking about permeabilized cell systems. Finally, you have to remember when you're studying mitochondrial bioenergetics in situ that when you're talking about whole cells and whole organisms even more, these are much more complex systems than just studying isolated mitochondria or mitochondria within permeabilized cells. So you have to remember that many phenomena that you might be measuring may not be generated by the mitochondria themselves. And we're going to see examples of that when we talk about measurements of membrane potentials in mitochondria in situ. But first, I want to talk about measurements of oxygen consumption. And here again, I want to enforce what I said before about if you have one measurement to do to look at mitochondrial bioenergetics in intact cell systems, measure oxygen consumption in these intact systems. Oxygen consumption is a quantitative measurement. As we will see, you can get a lot of information out of oxygen consumption measurements by using different inhibitors and looking at the results of these inhibitors. And finally, because it is a measurement of a direct consumption of oxidative phosphorylation, it's much less artifact prone than measurements that require probes such as membrane potential measurements that I'll talk about after we talk about oxygen consumption. Now, when you're measuring oxygen consumption in intact cells, Clark-type electrodes are perfectly fine. You can use Clark-type electrodes like we used for permeabilized systems. However, you do have to have more material just because the volume that's involved in Clark-type electrodes is a lot larger volume. Also, typically, you have to study your cells in suspension. Some cells do grow in suspension, and I recommend that you study these cells with Clark-type electrodes. But many cell types that we use in the lab are adhered cells, and these cells, when you suspend them, might have differences in their function. So Clark-type electrodes might not be the ideal way to study plated cell cultures. And in that sense, there are two types of experimental situations in which you can study these plated cells. One that is much less common are cell respirometers. So cell respirometers basically use a flow system in which the cells are plated inside a closed chamber, and there's flow of media over these cells and detection of oxygen concentrations both upstream of the cells and downstream of the cells. And this allows us to know how much oxygen is being consumed by these cells, and also allows us to add different substances to see how these cells respond to these changes promoted by these substances. Now, far more common than these kinds of cell respirometers are the seahorse measurements. So seahorse is a brand name, and it's a brand name of a specific system in which you're measuring oxygen consumption temporarily in a very, very small chamber. Basically, these are multi-well plates. They're either 24 or 96 well plates. The cells are attached to the bottom of the plates, and the lid of the plates contain detectors, both oxygen sensors and pH sensors, that are going to measure oxygen tensions and pH within the chamber. But the big secret of the system is that these measurement units on the lid of the system go up and down, and they do the measurements when they're down. And this decreases a lot the volume in which these measurements are done. Now, if your cells consume a certain quantity of oxygen in a very, very small volume, that's obviously going to change the concentration much more than this quantity of oxygen would change the concentration in a larger volume. So by changing this volume to a very small volume just while the measurements are being done, you can actually have a lot more sensitivity for plated cells. You can detect very small changes in oxygen consumption, very small amounts of oxygen consumed, because you're detecting them in a very small volume. So the change in concentration is going to be significant. This small volume is used only during the measurements. During the rest of the time in which the cells are incubated, you keep this lid higher up, and you keep the cells within the total volume of the media you're using. And that avoids a situation in which the cells could consume all the oxygen in that chamber and become anoxic. So the seahorse is really very good for detecting the small quantities of oxygen consumption of plated intact cells. I really recommend it for that kind of experiment. As in any other kind of experiment, you do have to normalize your experiment. You do have to work with known numbers of cells or with a known quantity of cell protein. Sometimes some people normalize their experiments by a mitochondrial marker because they want to look at oxygen consumption per mitochondrial mass and not per cell. That depends on what your question is and what you want to answer. This setup, the seahorse setup, can also be used for suspended samples. So you can either use suspended samples and not look at the effects of the smaller chamber, just measure the suspended sample in suspension. Then you will lose the advantage of the small chamber. Or you can actually stick whatever your suspended sample is, such as isolated mitochondria, to the bottom of these wells, and there are protocols published for that. I personally don't think that's the best idea if you have access to a Clark-type electrode, because you're changing your suspension. So if you have isolated mitochondria, they're normally in suspension. By plating them, you're adding a complicated factor to your experiment, which is sticking them there. Also, the consumables for the seahorse system are a lot more expensive than Clark-type electrodes that don't really have consumables. So if you have access to a Clark-type electrode, anything in suspension, including cells, I would do in suspension. On the other hand, for plated cells, especially cell cultures and cell lines, this seahorse system is really good because it really is sensitive. So it depends really on what you're using, what kind of equipment is best to measure oxygen consumption. Now, very typically with the seahorse system, because it changes the size of the chamber, you will not see traces in which you look at oxygen concentration changing over time, such as the traces that we saw before when we were looking at isolated mitochondrial systems. Typically, with this kind of measurement, what you will see is traces in which you look at the oxygen consumption rate or the respiratory rate over time. So this is the kind of trace that you typically will see. And there are very many different experiments that you can do in this setup looking at how the rate of oxygen consumption changes over time. The system actually allows you to make up to 4 additions of different things into your cell chamber during this time in which you're doing the experiment. Now, very commonly, 3 of these additions are going to be an addition of oligomycin, an inhibitor of the ATP synthase, an inhibitor of uncouplers such as FCCP, and then together 2 inhibitors of electron transport, which are rotenone, an inhibitor of complex 1, and antomycin, an inhibitor of complex 3. So let's quickly go over again what you would expect these drugs to do to respiration. So initially here we have a respiratory rate which reflects the normal consumption of oxygen by these plated cells. If you add oligomycin, if you add an inhibitor of the ATP synthase, you inhibit ATP production in mitochondria, increase the inner mitochondrial membrane potential because protons are not returning through the ATP synthase, and this decreases electron transport and oxygen consumption. So you're going to have lower oxygen consumption rates. If you add an uncoupler, this is going to decrease the membrane potential because now you're allowing these protons to return into the mitochondrial matrix freely. This liberates this thermodynamic barrier that was there for electron transport and oxygen consumption, and you're going to have a very high rate of oxygen consumption. Finally, by inhibiting electron transport in mitochondria with rotenone and antimycin, you're going to slow down oxygen consumption completely in mitochondria, and you're only going to have residual oxygen consumption, which is oxygen consumption from other cellular processes, which is not mitochondria. So this is an example just of those inhibitors. And these other three figures here are from the Nichols and Ferguson book. They're actually not of intact cells, they're of synaptosomes, but synaptosomes have reconstitution of the plasma membrane. So these are mitochondria within a plasma membrane, and very similarly they'll behave in terms of responses to stimuli to in situ mitochondria in intact cells. What they're showing here is that these mitochondria respond to different stimuli in the expected manner. So if you add, in addition to glucose, pyruvate as a second substrate, these cells, these mitochondria within these intact plasma membranes can respire more, can have more electron transport rate in the presence of uncoupler. If you add a substance that makes sodium enter these synaptosomes, this increases use of ATP in the synaptosomes and therefore increases respiration, but also limits respiration in the presence of uncoupler. This is expected. And finally, if you limit the entry of pyruvate into mitochondria, you also can have a limitation of the maximal respiration. So all of these are expected responses to these different stimuli. A very important point about the modulators you're going to use, and you're not only going to use oligomycin, FCCP, and rotenone antimycin, you might use other modulators too, is that you must titrate these modulators. Because typically these are substances that are membrane permeable, therefore lipophilic. They're going to accumulate in membranes, and if you use too much of them, they're going to have toxic effects. On the other hand, depending on what kind of media you have, the constitution of your media, if there's protein or not, if you don't have protein, the amount of effective oligomycin, FCCP, or rotenone antimycin might change, and also different cells respond to different quantities of these inhibitors. So it's really important that you titrate so you have the expected respiratory effect and you use the lowest amount of this inhibitor that generates the expected effect. So always titrate these substances. Having said that, although this oligomycin uncoupler and respiratory inhibitor experiment is a very common one, it's not the only experiment you can do under these conditions. You can also design experiments in which you inhibit the use of different substrates. You inhibit the use of fatty acids, or you inhibit the use of glucose, or you inhibit the use of amino acids. You can design experiments in which you look at the changes in pH and oxygen consumption with different inhibitors so that you can estimate how much ATP comes from glycolysis versus oxidative phosphorylation. And actually the Seahorse Company has a lot of experiments designed in this sense. You can look at those on their website. There are lots of different things that you can assess using these oxygen consumption measurements. We're going to stick to this basal oligomycin, FCCP, rotenone antimycin here because it is by far the most used experiment. And it's actually an experiment that tells you a lot about oxidative phosphorylation in situ. So let's look at the different values you get from this experiment. First of all, after adding rotenone and antimycin, you don't have mitochondrial oxygen consumption anymore. You inhibited complex 1, and complex 3. So oxygen consumption, electron transport is totally inhibited. If it's totally inhibited, the residual oxygen consumption that still happens here is non-mitochondrial oxygen consumption. This might be peroxisomes, it might be NADPH oxidases, it might be any source of enzymatic activity that consumes oxygen within a cell. But if you're looking at mitochondrial bioenergetics, typically you're going to subtract this value from all the other values in your trace, because this is non-mitochondrial oxygen consumption. So if you get this non-mitochondrial oxygen consumption and subtract that from your initial oxygen consumption, what you have is a value that we call basal respiration. This is the normal respiration of the cell under its normal conditions. This is how the cell is acting in its day-to-day life, in its normal media. So it reflects the use of ATP by the cell and also the coupling and efficiency of oxidative phosphorylation in the cell under normal conditions. When you add oligomycin, you can measure a value which is going to be the respiration that still is maintained even in the presence of oligomycin. So this is oligomycin insensitive respiration. This means that this respiration is independent of the activity of the ATP synthase, which was inhibited by oligomycin. Oligomycin insensitive respiration is a reflection of the proton permeability of the inner mitochondrial membrane. So you're going to have electron transport depending on how permeable the membrane is to protons, either through the membrane itself or due to proton transporting systems such as the mitochondrial uncoupling protein. So oligomycin insensitive respiration reflects the proton transport or the proton leak through the inner mitochondrial membrane. Oligomycin sensitive respiration is the rest of the respiration, the difference between insensitive and basal respiration. And this respiration typically is thought to reflect ATP synthesis in the mitochondria. So this is ATP-linked respiration. There's a caveat here that I have to say, which is that oligomycin insensitive respiration reflects proton permeability of the inner mitochondrial membrane, but it reflects maximized proton permeability. What do I mean by that? I mean that when you inhibit ATP synthase, you're going to increase the inner mitochondrial membrane potential. And when you increase the inner mitochondrial membrane potential, you actually increase proton leak. There's going to be more proton leak if there's more of a difference in proton concentration or in charge difference between the inner and the outer side of the membrane. So this measurement here is a maximized measurement of proton leak. It does not necessarily reflect the proton leak under basal conditions, in which the membrane potential is a little lower, therefore the leak is expected to be a little lower. That means that when you measure oligomycin sensitive respiration and say that's ATP-linked respiration, you're probably underestimating ATP-linked respiration a little bit. We know this, but this still is one of the best manners we have to estimate how much of respiration is due to ATP synthesis. We just have to keep in mind that we're actually maximizing proton leak when we add oligomycin. So these factors are changing respiration versus the basal respiration we had. Another value that you're going to manage to get from these kinds of traces is the difference in respiratory rates between basal conditions and conditions with uncoupler. When you add uncoupler, you're removing the inner mitochondrial membrane potential barrier to electron transport. So now all these components of the electron transport chain can transport electrons to oxygen, pumping protons against a very low membrane potential. So it's easy to transport electrons, and that's why you're basically maximizing respiratory rates. So the respiratory rates are expected to be higher under these conditions than under basal conditions. The difference between oxygen consumption under basal conditions and maximized respiratory rates is what we have as reserve capacity, as the ability to increase respiratory rates that the cell has. And this could reflect an ability to synthesize more ATP. And actually, reserve capacity has been linked under many situations in the literature to an ability to respond to stressful situations. Cells that have higher reserve capacity are more able to respond to stressful situations, probably because they can increase their ATP production more efficiently under this condition in which they need to increase their respiration and ATP production. Another value that you have when you add uncoupler is the difference between respiratory rates with uncoupler and respiratory rates with mitochondrial inhibitors. And that we call maximal respiration. In other words, the ability to reduce oxygen to transport electrons in the absence of an inner membrane potential. Here again, there's a caveat, which is that the mitochondrial membrane potential is not important only for ATP synthesis and not important only as something that limits oxygen consumption in vivo. The membrane potential is also important to bring substrates into mitochondria. We saw that in the last class. So when you add uncoupler, you may be limiting substrates entering mitochondria, and you may be limiting how many electrons actually reach the electron transport chain. If you have less import of pyruvate, you're going to have less oxygen consumption. So we call this maximal respiration, but we know that we might be underestimating maximal respiration because of this limit of the transport of these substrates into mitochondria. Another thing to keep in mind. So these experiments aren't perfect. They do have these slight problems, but if you know these problems, you can really get a lot of information from them. So let's talk a little bit about the kind of information you can get from changes in these different rates of oxygen consumption. So let's say you're measuring basal respiration in your cells and you see a change in basal respiratory rates. What does that mean? That can mean that your cell is using ATP at different rates. So for some reason, some process uses more or less ATP in this cell, and you're producing more or less ATP in mitochondria, changing the respiratory rate. Another thing it can reflect is a change in coupling. So if your mitochondria are more or less coupled, they're going to generate more or less ATP per oxygen consumed. So a change in basal respiration can reflect both a change in ATP use or a change in coupling. How do you differentiate if it's ATP use or coupling? You add oligomycin. So oligomycin-sensitive respiration is going to change, so that the respiration that decreases with oligomycin is going to change when the cell ATP use is changing. Okay, it's also going to change if the amount of ATP produced per oxygen is different. So it's also going to be reflective of a change in coupling. How do you differentiate if it's coupling or ATP use? You look at oligomycin-insensitive respiration. So oligomycin-insensitive respiration is not dependent on ATP use by the cell because ATP synthase is now inhibited. It's dependent on proton leak and proton transport. So this is going to reflect coupling of your mitochondria. And by analyzing the changes in basal oligomycin-sensitive and oligomycin-insensitive, you can tell if there's a change in ATP use or if there's a change in coupling in these mitochondria. Now, what happens if you have a change in reserve capacity? If you have a change in reserve capacity, either your cells are using more or less ATP, or they're making ATP with more or less ability, there's a change in coupling, or you have a change in the maximal electron transport rate. How you can differentiate these different conditions, first looking at oligomycin-insensitive respiration, and second looking at the changes in maximal respiratory rates. So if you have a change in maximal respiratory rate, you probably have a change in the ability to reduce oxygen and electron transport capacity. So with one trace, with 3 different additions, you could come to the conclusion about the whole function of the mitochondria. How much ATP is being synthesized, how coupled these mitochondria are, and how much electron transport capacity these mitochondria have. It's actually a really good way to look at mitochondrial bioenergetics in intact cells. Some things I want to comment about cell respiration experiments. First of all, and the makers of Seahorse tell you this all the time, you do have to do initial titration experiments. First, titrate for cell quantities. You want a cell quantity in which you can detect changes in oxygen consumption well, but they're not saturated, so if you have a change in which you have an increase in respiration, you can still detect this change. And this really requires titration for every single cell type. So you have to do these preliminary experiments and make sure that your cells are well distributed and that you have good measures of oxygen consumption under your conditions. A second thing is that you really have to be careful how you plate these cells within the wells. You want the cells plated centrally. You don't want cells on the periphery because they can actually get stuck there and they can actually not be measured as the oxygen electrode goes up and down. So you don't want cells that are on the wall on the sides. You want cells that are on the bottom of the plate, and plate them in the very center, and that will help with that. I already talked about this. You really need to titrate oligomycin or uncoupler. Otherwise, you might have toxic effects of these modulators. This is very important. Rotenone and antimycin you want to add in quantities that are really going to inhibit the whole of mitochondrial electron transport. So titration of those two is less important. But oligomycin and uncoupler, the ideal amounts will change for every cell type, and many times within the same cell type under different conditions. So you really need to titrate to have good experiments, to have trustworthy data. Another thing is always recommended is to spread duplicates or triplicates of the same experiment across this plate and not keep them just on one side, just in case you have differences in measurements between the different detector systems. Of course, you're going to have either 24 or 96 different detectors in your system. And then the things that we talked about, that our measurements can be altered by the addition of oligomycin and uncoupler themselves. So when you add oligomycin, you're increasing the membrane potential, and by increasing the membrane potential, you're maximizing the proton leak. Therefore, when you look at the proton leak as oligomycin insensitive respiration, you're actually looking at a maximized proton leak. And the real proton leak under basal conditions is probably lower than that. So keep that in mind when you're analyzing your data. Another thing that we talked about is that maximal respiration may be underestimated, because when you add an uncoupler, you're decreasing the membrane potential and also decreasing the uptake of respiratory substrates, such as pyruvate. It is very common for some authors to add the FCCP or other uncouplers together with pyruvate to get better maximal respiratory rates. I strongly recommend that. That actually improves your electron transport and gets you closer to maximal respiratory rates. But even by adding more pyruvate, you still may have a limitation in uptake of these substrates because of the low membrane potential. In fact, Roger Castillo's group published a paper that showed that if you add uncoupler with or without pyruvate, after adding oligomycin, you may underestimate the maximal respiratory rate. So if this value of maximal electron transport is really important to you, I would consider doing an experiment with uncoupler and without oligomycin, and doing the oligomycin experiments in separate wells so that you really have a trustable maximal respiratory rate. Finally, you have to remember that ionophores act on all membranes. So uncouplers are going to uncouple plasma membrane potentials too. And plasma membrane potentials can affect ATP levels in the cell and can affect mitochondria. So you do have to take that in consideration, especially in cells that have significant changes in plasma membrane potentials under your experimental conditions. Finally, we talk a lot about mitochondrial function and looking at respiration because we're people who are interested in mitochondria here, but do not overlook non-mitochondrial oxygen consumption. So if you have a significant change in non-mitochondrial oxygen consumption, that can indicate a number of changes in these oxygen-consuming pathways in the cell, which might be interesting to you and which you might want to have a look at within your experimental paradigm. So that's what I wanted to tell you about cell respiration experiments, oxygen consumption in intact cells. I'm going to come back in the next video and talk to you about how to measure inner mitochondrial membrane potentials in intact cells. So till then.